Hello and welcome to Chapter 25, Seedless Plants. So we begin in this chapter our study of plants, and we start with seedless plants. Now, seedless plants include things that you're familiar with, mosses and ferns. And while seedless plants make up only a small fraction of plant life today, 300 million years ago, seedless plants dominated the landscape. Now, seedless plants require moist habitats, as we'll see, in which to grow, and the early earth was ideal for that. It was full of water, swampy habitats everywhere, perfect for the growth of seedless plants. Now, as seedless plants complete their life cycle and they die, the decomposition of them created large deposits of coal, and that's the coal that we mine today. And it's these deposits of coal which gives the carboniferous period its name. So plants, in order to leave the water and transition onto the land, had to overcome a number of hurdles, including desiccation, radiation, and gravity. And so in this chapter, we'll follow the journey of green plants from the water to dry land. So our objectives for this chapter include describe the adaptations that allow plants to colonize the land, describe the traits shared by green algae and land plants, explain why charophytes are considered the closest relative to land plants, identify the main characteristics of the bryophytes, describe the distinguishing traits of liverworts, hornworts, and mosses, but really we're going to be looking at mosses. Identify the new traits that first appear in seedless tracheophytes. Discuss how each trait is important for adaptation to life on land. Describe the life cycle of a fern. And explain the role of seedless plants in the ecosystem. Okay, so here we go. In the beginning, so there are more than 390,000 species of described plants. 94% of them, about 366,000, are seed-bearing plants. The remainder, of course, being seedless plants. Seedless plants, we call them bryophytes, arose from algae, specifically green algae. Those are the charophytes, about 400 million years ago. So we can see green algae down here is a typical charophyte living in the water. You can see it kind of looks a little tree-like, looks like a little tree. So there is some appearance here that is very familiar when we start looking at terrestrial plants. Over here we have a bryophyte, which is a typical moss. This is a mat of moss on some rocks. Moss you'll find in certainly moist habitats, especially on like dead logs. Moss grows on rocks. Moss grows along creek banks. You'll see a lot of moss. So living in water is relatively easy compared to living on land, as we'll see. Now, aquatic algae, the charophytes, had to make some drastic changes in order to survive terrestrial habitats, in order to become bryophytes. So in order to successfully conquer the land, plants had to successfully overcome several obstacles, which I mentioned in the introduction, namely desiccation, radiation, and gravity. So desiccation is a process of drying out. Okay, so you know when you buy a new electronic device, inside the box usually there's a little pouch, a little satchel, and if you shake it, kind of sounds like there's grains of rice in there or little grains of sand in there. It's called a desiccant. Sometimes when you buy new clothes, if you reach into a pocket of a new garment, you'll find one of those little things. Or even if you buy food online, when the box shows up, sometimes it'll have that little satchel in there and you shake it. It sounds like sand in there. That's a desiccant. That keeps whatever is in the container or in the pocket of your clothing, that it absorbs moisture. it dries things out. That's what desiccants do. So desiccation is a process of drying out. Now, being out of water means that in order to survive on land, plants had to learn to retain moisture, because once you leave the water, right, you're no longer wet. So, but as you know from last semester, all organisms are essentially we have more water in us than we do anything else. Water is essential for survival of any organism, including land plants. So in order to leave the water and survive on land, plants had to learn how to retain moisture. We'll look at that process. Radiation. Now, being out of water means that plants had to learn to protect themselves against UV radiation. Water provides a filter that keeps ultraviolet radiation, the damaging radiation, the mutagenic radiation, out and away from you. Now, once plants leave the water, that protection of that layer of water is no longer there. So plants had to learn how to protect themselves, had to evolve to protect themselves against damaging radiation. And gravity. Water provides buoyancy. Plants don't have to be structurally tough, because water provides buoyancy. That's why astronauts train in a giant swimming pool, because it allows them to be buoyant. It's like being in microgravity. There's no weight. In order to leave the water and survive and thrive on land, plants had to evolve structures that would allow them to stand up, literally stand up, against the force of gravity. So some of the adaptations that helped green algae become terrestrial plants was this thing called alternation of generation. Now, this is a life cycle in which the plant cycles between the sporophyte and the gametophyte. Now, you're already familiar with those terms. It's a reproductive strategy. It's a life cycle in which the plant, and we'll see with once we get to real true land plants, sporophyte is the dominant generation. You know that sporophyte is 2N, or is diploid. And then the alternation of generations means that the organism goes through the sporophyte phase, and then it goes through the gametophyte phase. And you know that a gametophytes produce gametes. Gametes are haploid, or 1N. So the gametophyte is 1N. And so this alternation of generations means that the organism rotates between a diploid sporophyte and a haploid gametophyte, back to a diploid sporophyte, then back to a haploid gametophyte. Apical meristem. Now, apical means at the apex, or at the tip. These are specialized areas in roots and stems where cell growth occurs. Rapid cell growth occurs in meristematic regions, and meristematic regions occur at the apex of roots. That's the tips of roots, and at the tips of shoots. We'll look at that closely in a minute. Cuticle. Cuticle is a waxy outer layer that prevents the loss of water. So the cuticle on leaves, you'll be familiar with that when I show you, prevents desiccation. Now, cell walls. Plants have evolved cell walls, like bacteria evolved cell walls, and in those cell walls is this component called lignin. We say that the cell walls of plants are lignified. This lignin is a rigid component that allows land plants to defy gravity, gives them strength, and thus allows them to grow higher and higher and higher. And for instance, you know, a giant redwood tree doesn't blow over in the slightest wind because its cells are full of lignin. And then flavonoids. Flavonoids are pigments. You're familiar with For instance, the orange of carrots or the reds of apples. Those are flavonoids, but those pigments absorb ultraviolet light and therefore protect the inner structures, the DNA, the genetic material, from being damaged by ultraviolet light. And so here we see real quickly the alternation of generations, where we go from the 1N phase to the 2N phase, or the gametophyte phase, 1N, to the sporophyte phase, 2N. We'll talk about the apical meristem or the meristematic regions. This is a root tip, an onion root tip, and this is an area of really rapid cell growth. Here is, you can see the shiny layer on this ivy leaf. That's the cuticle, that's the waxy cuticle. And here we see, this is bamboo, and some bamboo can grow 30 feet high, but it's lignin in the cell walls of these plants that allow them to defy gravity and grow so high. And finally, we have the colorful fruits and vegetables. Those are all flavonoids that actually protect plants from ultraviolet light. Okay, so we'll look at these one at a time. Here's alternation of generations. And we talk about land plants, most of what you're going to be familiar with. When you look outside and you see, we'll talk about ferns. When you look outside and you see a fern, or you watch a Discovery Channel and you see ferns in the jungle, or maybe even you or your parents have ferns in pots hanging on your porch. Lots of people do that. Ferns are decorative plants. That's the sporophyte generation. That's the 2N or the diploid generation. Now, sporophytes, as you know, and anyway, based on the name, you can figure out that they make spores. Sporophytes make spores. And so they make spores, and some of these spores are going to be male, and some of them are going to be female, or some of them are going to be plus, and some of them are going to be minus, however you want to look at it. Now, those spores, okay, so to produce spores, meiosis is the process, and spores are 1N, they're haploid. So we go from a diploid sporophyte to haploid spores. Now, those spores will go through mitosis, and some of them will become male gametophytes, okay? Remember that gametophytes make gametes, and gametes are 1N or haploid. So gametophytes are haploid. So some of those spores will turn into male gametophytes, if you will, and some of them will turn into female gametophytes. Females produce eggs or ova, males produce sperm, those are the gametes, respectively. And so sperm is 1N, egg is 1N. When sperm meets egg, fertilization occurs, and fertilization creates a zygote, which is diploid. So now we're back to the 2N generation, or the diploid generation, and that zygote grows into a new sporophyte, which is 2N. Sporophytes make spores, and now we're back into 1N. Spores make gametophytes, zygote, back sporophyte. Okay, so it's alternation of generations. 2N to 1N, back to 2N. Sporophyte to gametophyte, back to sporophyte. That's alternation of generations. That allowed plants to better survive and thrive on land. So sporophytes are 2N. Sporophytes make spores. Microspores are the male version. That's the little arrow. And megaspores are the female version. That's the plus, because males give, females receive. And so sporophytes are heterosporous. Heterosporous means that one sporophyte makes two different types of spores, both plus and minus, or both male and female. So spores are 1N. Spores are haploid, and they undergo mitosis to produce female and male gametophytes. Gametophytes make gametes. So gametophytes are 1N. They make gametes, also 1N, which are reproductive cells. And gametes, we have plus and minus, or female and male gametes fuse to form the zygote. Now we're back to the 2N phase of the life cycle. The zygote develops into a new sporophyte. So now we're back into sporophyte. So you can look at that as an alternation between 2N and 1N, back to 2N, or sporophyte to gametophyte, back to sporophyte. Either one of those is fine. When you think about alternation of generations, both of them are true. Here is a little moss down here on some dead wood. And I call these little things aerials. They're called setae. And on the top are these little sporangia, these little like balloons, almost looks like a balloon on a string. And these little balloons, that's where they're sporangia, so that's where you find spores. And so these little spore balloons, right, will dry up and burst open, and the spores get spread by the wind, distributed by the wind. Some of them are male, some of them are female, and that's how this organism disseminates itself in the environment and propagates its species. Okay, that's alternation of generations. That helped plants leave the water and survive and thrive on land. So the apical meristem is an area of rapid cell division. That's mitotic cell division. You have meristematic regions in the tips of roots and in the ends or the tips of stems. And that allows plants to grow rather quickly. And so, if you have roots that are growing, pretty fast, digging down into the soil, that's going to help that plant retain its position in that habitat, in that ecosystem, right? So that's going to help a plant to stay fixed in the environment or remain on land. That's one of the benefits of apical meristematic growth, is it allows plants to, again, maintain their position on land. That's the whole point, right? We're evolving from the water to the land. So as new cells elongate in the meristematic regions, the stems grow higher and the roots get deeper, and that gives plants access to additional resources. As the stems grow higher, the plants have more access to sunlight, and as the roots grow deeper, they have more access to water and to nutrients from the soil. So here is a diagram, that illustration here, that shows you a typical plant. Now the stem, here's the stem, and don't think of stem as just like the stem of a rose, but the trunk of a pine tree, that is the stem, okay, of that plant. So here we see the stem, and down in the ground we see roots, and at the very tips of roots are the apical meristematic regions, or the regions of very fast mitotic growth. And then up here, the tips of the apex, the tips of the shoots, stems, branches, if you will. As they branch off, we're going to have meristematic regions there as well. Those are going to be regions of very fast growth. You do not have meristematic regions in leaves, okay? So here we see from your text the illustration of a root tip, and there's the area we call the apical meristem. It's the meristematic growth or mitotic growth at the apex. That's the apical part, at the apex of the root, or on the reverse side of the coin, the apex, the tip of the shoot. And in the lab, when we do this class live in the lab, you look at a slide on the microscope, or you prepare a slide yourself of an onion root tip, which you see here, and you see all these little purple dots are nuclei of cells, and in this region you see all sorts of different phases of cell division, like this. Okay, you stain the sample, the specimen, and you can see all different stages. And when you look at this, it's not just all prophase or not just all metaphase. You see metaphase over here, and you see anaphase over here, and you see telophase and prophase, and I mean, you see it all. And so from this slide, just one little sample. I mean, here you see clearly telophase. Here is anaphase going on here. I don't see any metaphase here. This is like early anaphase. But here you can see there's no plasma membrane, right? And these cells are a little bit smaller than, say, this one, right? And there's the cell wall forming. So this is cytokinesis going on here, separation of cells. There's a cell wall forming. Eventually, the cell nuclear membrane will form back here. And here, in fact, here are two daughter cells, right, that are just fully separated. See how much smaller these cells are than this one, for instance. But these will grow, right, to reach this size, the adult size, but these are two new daughter cells. Okay, so you see all of that in the apical meristem of a root tip or a shoot tip. Rapid cell growth, and you see all stages there. Okay, one of the other things that allowed land plants to thrive, water plants that thrive on land, is the evolution of the cuticle. Now, cuticle is a waxy, waterproof coating on leaves and stems, prevents desiccation. Now, the only problem is it prevents water loss, and so at the same time it prevents carbon dioxide, in the case of leaves, carbon dioxide from getting in. And you know from your study of photosynthesis, carbon dioxide has to get into the plant, right, in order for the plant to make sugar to make energy. That's the whole bottom line. But the cuticle, this waxy waterproof coating, prevents carbon dioxide from diffusing in. So the plant has evolved these tiny structures, tiny cells called stomata. Stoma is singular, stomata is plural, and these stomata are just little modified cells that have a pore in them, that are pores that allow the absorption of carbon dioxide. We see them abundantly on the bottoms of leaves, and again in lab, we go out and we pick a leaf and we bring it back in and we flip it over and we put a little nail polish, clear nail polish on the bottom. We let that dry, peel it off, put it under the microscope, and you can see the pores. You can see the stomata on the underside of that leaf. It's a really, really cool exercise. But they allow carbon dioxide to absorb into the leaf in spite of the cuticle. So here we see these beautiful southern magnolia leaves. You might have one of these trees in your yard. And this shiny coating, that's the cuticle. You can actually go take one of those leaves, and with your fingernail, you can actually scrape off some of that wax, some of that cuticle. And on the underside are these, now this is very close up under the microscope, but you have these specialized structures, 1, 2, 3, called stomata. These pores, they're openings. These pores are closed or mostly closed. And these cells around them are specialized cells. They're called guard cells. And when the plant needs carbon dioxide, the guard cells open the pore up and carbon dioxide gets absorbed in. And then at night, when the plant doesn't absorb carbon dioxide, the guard cells close the little pores. But these are the stoma that allow carbon dioxide in spite of the cuticle. Another evolutionary advancement that allowed plants to survive and thrive on land. We have lignin. Lignin is a waterproof polymer. It's deposited in plant cell walls, and it provides strength and durability. And we say it's the backbone of the plant. There is no backbone per se, but that's why it's in quotation marks. But it's the backbone of the plant. It's the strength of a plant. So when you think about that 300-foot high redwood in Washington State, USA, that's lignin that is allowing that thing to grow that high. But there's lignin in a rose bush, and there's lignin in a wildflower stem, okay? But the higher you go, or if you're a plant, the higher you go, the higher you want to go, the more lignin you need to have. So woody stems, like trunks of trees, are full of lignin, and also it protects this lignin, which makes these woody stems, protects against insects and disease. Now, lignin is abundant in xylem. That's one of the vascular tissues. I mention it here. We'll talk about it in a little while, but xylem is a vascular tissue, and lignin is abundant in xylem, and it prevents, lignin, by the way, prevents water loss during transpiration. So here we see these nice tall tree trunks full of lignin. That's how they're able to grow so tall. And here's a little illustration showing transpiration. You don't need to know about transpiration at this point, but transpiration, the idea here is water gets absorbed in the roots, it travels up through the stem into the leaves, and then it gets exhaled, if you will, transpired out of the leaves and into the environment. Okay, so water comes up the plant, the plant uses the water that it needs, and the excess gets water vapor gets released from the plant. These stems are full of lignin, which helps prevent the loss of water as it travels up through the plant. That's the whole idea behind lignin and transpiration. All right, that's the connection. All right, quick check. Which of the following statements about plants, just in general, we're talking about plants in general, is true? It says check all that apply. Choices are: lignin provides strength and durability to tree trunks. The cuticle is a waxy coating on stems and leaves. The cuticle prevents desiccation. The apical meristem is an area of rapid cell growth on the tips of leaves. And sporophytes are diploid while gametophytes are haploid. It says check all that apply. We'll look at them all individually. So lignin provides strength and durability to tree trunks. We just talked about that. And that 300-foot redwood in Washington State can grow so high because it has lots of lignin. So we know that's true. Check all that apply, right? The cuticle is a waxy coating on stems and leaves. We just talked about that, and I showed you the picture of the magnolia leaf with the shiny cuticle. So the cuticle is definitely a waxy coating on stems and leaves. Check. The cuticle prevents desiccation. Well, that is one of the purposes of the cuticle as well as protection, but to prevent water loss. That's desiccation, drying out. So the cuticle prevents that. The apical meristem is an area of rapid cell growth on the tips of leaves. Well, the apex meristematic growth regions do not occur, as I mentioned, in leaves. They occur at the apices. And where do you find apices? At the tips of roots and at the tips of shoots. So apical meristem is an area of rapid cell growth, yes, but it's not on the tips of leaves. And then sporophytes are diploid while gametophytes are haploid, and we know that's alternation of generation, so that's true. Let's go to the video. This is a really good video by Wise Guy Science about seedless plants. Good for you to watch, not very long. Come back. So here is a nice little illustration from your textbook showing you the flow of the evolutionary process of going from the water to the land. We start here with the charophytes. Now the charophyte that we're going to deal with, there's lots of different charophytes, but we like the green algae because it's the green algae that gave rise to land plants. All right, so we'll talk about green algae. Now, green algae gave rise to the non-vascular seedless plants. We'll talk now, we call them the bryophytes, and we'll talk about moss as our indicative species of bryophyte, but that's a non-vascular plant. Remember, I mentioned xylem, which is a vascular tissue. The other vascular tissue is phloem, and so non-vascular plants have neither of those tissues. So green algae, non-vascular, of course, but the transition here is to seedless plants, non-vascular. Okay, now seedless plants, non-vascular, gave rise to the vascular plants. You see the evolutionary progression. So we have green algae in the water. Now we're going to come out onto the land as a non-vascular seedless plant like a moss, and then evolutionarily, in that evolutionary time, we're going to transition into a vascular seedless plant. And we're going to use fern as our representative organism from the vascular seedless plants, and from the vascular seedless plants we're going to evolve into the seed-bearing plants. There's a train. I'm going to pause. Okay, so ferns evolved into, or vascular seedless plants like ferns evolved into the seed plants, which is chapter 26, which we'll certainly talk about later. All right. So green algae is our representative charophyte that's going to lead us into land plants. So like land plants, charophytes, like green algae, their cells divide along cell plates. You'll recall cell plates from our study of cell division in 101. Now, like land plants, charophytes, like green algae, exchange materials with adjacent cells via plasmodesmata. Those are just little channels that form between adjacent cells so they can exchange nutrients or exchange water. And like land plants, charophytes, like green algae, demonstrate apical growth. So that's the apical meristematic growth. We see that in green algae as well as land plants. So that's an evolutionary advantage that carried over from the green algae to the land plants. And so here we see a typical charophyte, and you can see it kind of looks like a little tree, and you can see the tips of these little stems, which you can imagine meristematic, very rapid mitotic growth occurs. And here is green algae. Someone's holding it. You can see the size of these little plumes here compared to this person's hand. So, you know, they're small. These are the kinds of plants that you might even see in your aquarium, or if you go to Red Lobster and you look in the aquarium, you'll see these kinds of plants, okay? Okay, now, Charophytes, like green algae, lead us into the bryophytes, which are non-vascular seedless plants. These are the closest extant relatives to early terrestrial plants. Extant meaning living, as opposed to extinct, which means passed away many years ago. So these are closest living relatives to early terrestrial plants. Most species live in moist environments. Again, we've just, as a bryophyte, we've just come out of the water. So we still have, we still have a need for water. Our sperm have to swim, and they don't swim on dry land, all right? They swim in water. So we still have a need to be in a moist environment where there is water so that we can reproduce. I'm talking as a bryophyte. But there is no lignin, and so bryophytes do not grow very high. And so our representative organism, bryophyte, is going to be the moss. And if you've gone walking through the woods, or maybe even in some area, moist, shadowed areas of your backyard, you might see moss. You can look at it. It doesn't grow very high, maybe a quarter of an inch above the ground. It cannot grow very high because there's no lignin in their cell walls. And they are non-vascular. There are no vascular tissues, namely xylem and phloem. And the bryophytes include liverworts. I got a picture. The bryophytes include liverworts, hornworts, and mosses. Liverworts, they're called liverworts because this is called the thallus, and the thallus portion of the organism looks like the liver, right? These lobed structures look like the lobes of the liver. That's what they say. That's where it gets its name, liverworts. And then you have the hornworts. Here's the thallus down here, and you have these little areoles that grow up, and on the end of the areoles are the sporophytes, and they kind of look like, they're kind of horny, right? They kind of look like little horns. That's where it gets its name. But our representative bryophyte is going to be the moss, and you've seen this before, I'm sure. If you haven't been out into the woods, gone on a hike, get out into the woods this spring, go on a hike, and look at some moss. But here we see some dead wood, and moss grows on dead wood. And again, they don't get very high because there's no lignin. All right, so let's talk about mosses. This is the most numerous bryophyte. There's about 10,000 species. They inhabit the tundra, everywhere from the tundra to the tropical rainforest. You see them, again, there's going to be some moisture available. Mosses need that. And in the tundra, in the warm part of the year, in tundra, things thaw out, because tundra, I think you know, tundra is frozen, right? It goes very deep. The ground is frozen all the time, except there is a warm period with regard to tundra during some portion of the year, and it depends on where you are, Siberia or Alaska. Depends on where you are as to how long that warm period lasts. But in that warm period, things thaw enough for pools of water, or for water to pool around. And that's a great place for mosses to thrive, at least for a portion of the year. Certainly in tropical rainforests, there's water all the time, and so you have moss all the time. Okay. The rhizoid is the, I say, root-ish structure that anchors the thallus to the substrate. I say root-ish because it's not a root as we know roots. It doesn't go, rhizoids do not go deep into the ground. They do not anchor strongly the thallus to the substrate, the ground or the rock. And that's why it's easy to go into the woods or wherever, your backyard, if you have it, and you can just reach up under a mat of moss and just peel it up. You can do that because there are no roots. It's just a rhizoid that kind of gently adheres the moss to the substrate. Okay, Mosses provide food and shelter to small animals and even provide food to big animals. Mooses eat mosses, as do white-tailed deer here in Virginia. Mosses are sensitive to air pollution, so they're great indicators of ecosystem health. Now, the gametophyte, with regard to mosses, is the dominant phase of the life cycle. Now, gametophytes make gametes, right? So gametophytes are 1N or haploid. Male gametes and female gametes, we call them gametangia, develop on separate gametophores. Now, those gametophores, we call them antheridia. These are the male structures which produce sperm. Antheridia. How did I keep this straight in my mind as a young undergraduate? Anther sounds like antler, and male deer have antlers. Antheridia. Okay, that's how my mind works. That's how I remembered. But antheridia are the male structures that produce sperm. Archegonia are the female structures that produce ova or eggs. And so you have these two separate structures developing on the same gametophyte, by the way, but two separate structures, one male, one female, as opposed to the sporophores, right, in algae, which produce both male and female. Fertilization then occurs in the archegonium. So we have the archegonia is plural, archegonium is singular. Same thing, this is the female structure. That's where fertilization occurs. And so the sperm, again, which are 1N, have to swim. They have little flagella, just like human sperm, and they have to swim to the egg, which is tucked down in the archegonium. So it swims to the egg and then fuses with the egg, forming the zygote. 1N plus 1N is 2N, so the zygote is 2N, and the zygote develops into a new sporophyte. Sporophyte is 2N. Sporophyte gives rise to male and female gametophytes. So now we're back to the 2N generation. So we went from 1N to 2N, gametophyte to sporophyte, back to gametophyte. That's alternation of generations. So here we see an illustration. This is similar to the one in your textbook. Alternation of generations in mosses. Okay, so here we see let's start over here. Here we see the gametophyte generation. Here's the male gametophyte, 1N. Gametophytes are always 1N, haploid. Here's the female gametophyte. You notice they're on separate structures. Females make eggs, males make sperm. Here we see the little 1N sperm cells swimming over to the archegonia, and the eggs are tucked in. That's the little dark green dots. The eggs are tucked in to the archegonia. You see here, eggs are 1N. Sperm fuses with egg. 1N plus 1N equals a 2N zygote. So I should say sperm here fuses with egg. 1N plus 1N yields a 2N zygote, still in the archegonium. That's the process of fertilization. And that egg develops into a new sporophyte. So here we go from 1N generation to the 2N generation, or gametophyte generation, to the sporophyte generation. And that's the alternation. So it's sporophyte to gametophyte back to sporophyte, or 2N to 1N back to 2N. Sporophytes make spores. Spores undergo meiosis. Spores are 1N. They undergo spores are 1N. They undergo mitosis to form the new female and male gametophytes. All right. Oh, yeah. This area is called the venter. You might see that in your review exercise. That's called the venter. It's like a vent, right? That houses the egg, the ovum, and that's the little area that the sperm has to swim down in. It's called the archegonium. And that's called the venter of the archegonium. All right. Not that important. All right, so this is a really cool video by Science by the moss life cycle under the microscope. It's narrated. It's only a couple minutes long. Please watch it. It's all real life stuff. Okay, no animations here. All right, quick check. Which of the following statements about the moss life cycle is false? Your choices. The mature gametophyte is haploid, or 1N. The sporophyte produces haploid, or 1N spores. Fertilization occurs in the antheridium. The zygote is protected in the venter. All right. Which one is false? Well, look at them individually. The mature gametophyte is haploid, or 1N. Gametophytes produce gametes. Gametes are 1N, so gametophytes are always 1N. Sporophyte produces spores. Spores are haploid, 1N, so that's true. Fertilization occurs in the antheridium, right? In my mind, the antleridium, which is male, fertilization occurs in the female, in the archegonium, so we know that that is not true. And the zygote is protected in the venter. Remember that slot, the venter of the archegonium is where you find the zygote, the egg, and ultimately the zygote. So fertilization does not occur in the antheridium. It occurs in the archegonium, and so C is false. Another video, this is from Bozeman Science, about plants. It's just all about plants, and it just walks you through from the green algae to the land plants. It's just a nice little overview as we proceed into the seedless plants. Okay, so remember, back to this chart. Remember now, we've gone from the charophytes, right? Our representative organism was the green algae. Green algae gave rise to the non-vascular seedless plants, or the bryophytes. Our example here of the non-vascular seedless plant was the moss. We looked at the moss and its life cycle. The non-vascular seedless plants gave rise to the vascular seedless plants. Includes lots of different things. Vascular seedless plants, but our example is going to be the ferns. We'll look at that in just a minute, and the ferns, of course, gave rise to the seed-bearing plants. All right. Okay, so we've gone from the charophytes, the green algae, to the non-vascular seedless plants by way of the mosses. All right, and so now we're going to evolve into the vascular seedless plants. Vascular tissues allowed plants to move further into land and more dominate terrestrial habitats. So there are more than 260,000 known species of vascular plants. We call them tracheophytes. That represents about 90% of the Earth's vegetation. Most of what you see, well, can't say that. So we call them bryophytes. They successfully transitioned from water to land. They still need water, right? Their sperm still need water in order to swim to the egg, to the ova. Now, one of the benefits, or one of the evolutionary adaptations to moving further into land, was the evolution of vascular tissues. Now, think of vessels, like blood vessels, like your arteries and your veins, right? That's called your vascular system. So think of vascular systems like your arteries and veins. They are vessels that travel through the organism that carry nutrients and carry water and carry all sorts of things, just like your blood vessels do. In plants, we call them xylem and phloem. It's two types. And the evolution of vascular tissues was certainly a major factor in plants conquering land. Now, vascular tissues provide conduits for the transport of water and minerals and sugars throughout the plant. Development of sophisticated root systems was critical for land plants, and roots are also vascular, and the vascular presence allowed those rhizoids to evolve into root systems for land plants. All right. And then the development of leaves, which also contain vascular tissue. These are organs of photosynthesis. The development of leaves for absorbing light energy was critical to plants moving further in onto land. Vascular tissues, wait, what? Well, vascular tissues evolved around 430 million years ago, and in fact, fossils from that time show clear evidence of xylem and phloem. Those are the two vascular tissues, like arteries and veins, xylem and phloem. So xylem provides storage and transport of water and nutrients throughout the plant. Okay, these nutrients flow upward through xylem, upward from the roots, where water and nutrients are absorbed from the soil, to the stems and the leaves. The cells of xylem are called tracheids, and they are lignified, all right? That's where you have the cell walls of tracheids, which are the cells of xylem, are fortified with lignin. Remember that lignin provides strength and support, allows stems to grow to great heights. And height here is a selective advantage, right? Because tall plants are better able to absorb sunlight. If you're a short plant and you're down in the understory of the forest, you don't get much light. Trees that spread out in the rainforest, for instance, way at the top, they get the most sunlight. All right, so height is a selective advantage for absorbing sunlight. Spore and seed dispersal. So if you're a seedless plant, you're going to make spores, and the higher you are, the better able you are to release your spores, and they get carried by the wind to other habitats. We call that seed dispersal. And high plants or tall plants outcompete shorter plants for resources like sunlight and like wind. And the higher the plant, the deeper the roots, and so you have better access to water and nutrients than shorter plants. Okay, vascular tissues. The other vascular tissue is phloem. Vascular tissue that transports sugars and proteins throughout the plant. Now remember that glucose is made in the leaves. That's where photosynthesis mainly takes place, in the leaves. The leaves absorb carbon dioxide and they mix with water and you make glucose, sugar, and then that sugar flows to all parts of the plant, whereas water gets absorbed from the ground and flows upward to all parts of the plant. Okay, so water travels in xylem upward, and sugars and other things travel from the leaves through phloem downward. Okay, here's the way my mind works. The way I remembered this is, you know how water, if you have a creek, it starts in the mountains, flows down to the ocean. See, it flows downward. It flows with gravity. So sugars and proteins flow with gravity down to all parts of the plant. They phloem, right? You flow them down to all parts of the plant, whereas xylem, water, it's the opposite side of the coin. Water travels upward through xylem. Sugar travels downward, flows downward through phloem. It's the way my mind works. Sieve elements are the conducting cells of phloem. So we have tracheids in xylem, we have sieve elements in phloem, and the cells in phloem are kind of continuous, but they have these, porous walls between the cells, and we call those porous wall sieve plates. All right. So this is just a cross-section of a leaf. We have both xylem and phloem in leaves, as well as in stems, as well as in roots, as we'll see in a second. So this cross-section of a leaf, it's not a great illustration, I know, but here, this is called a vascular bundle. A vascular bundle is xylem and phloem all together, right? Because it's like arteries and veins, right? You have the femoral artery in your leg, but alongside of it, you have the femoral vein. Artery goes one way, vein goes the other with blood. So it's like that. You have xylem going one way, phloem going the other, but they're side by side. And so here you see xylem, in this case, the large openings represent xylem, the smaller openings represent phloem. And so here you see a number of vascular bundles. So this is a cross-section of a leaf, right? So this would be, you see how much bigger it is? This is like the main vein of the leaf. And these are the accessory veins. I'll show you a leaf here in a second. So vascular tissue. So here's that main vein of the leaf where you have vascular bundles. This is all one continuous vascular tube. In cross-section, you just see it as a little bundle there. We call it a vascular bundle. And these are the accessory veins. Again, xylem and phloem together. And then even off of those, you can see these even tinier accessory veins. Again, xylem and phloem. Here is a nice illustration showing you the different distribution of xylem and phloem in roots, stems, and leaves. Now, what's going on in the root? Well, you're absorbing water and nutrients, right? You're in the ground. You have no need for photosynthesis. You're in the ground. You're a root. You absorb water and nutrients, and you need to send that to the upper part, the above ground part of the plant. Most of what is going to be there, would you think, xylem or phloem? Well, xylem carries water and nutrients upward. So most of the root, as you can see here, most of the vascular tissue in the root consists of xylem. As we get into the stem, we sort of see an even distribution. These are your vascular bundles out here, consisting of xylem and phloem, kind of equally distributed. And then when we get into the leaves, there's your cut. This is a cross-section of a leaf. That central vein is going to be a large vascular bundle, and these accessory veins are going to be a little bit smaller, as we saw in this leaf over here. And microscopically, again in the live lab, you would be looking at this slide in the microscope, and this is a core or pith, depending on which type of plant you're looking at. But these are vascular bundles right here. They're vascular bundles. This is xylem, the large holes. This is phloem here. And this is just support. These are cells that just support the vascular bundles. They're called sclerenchyma. Certainly don't have to remember that. But this is a vascular bundle. And here you recognize this. This is a stalk of celery, but you can do this at home. You can take a stalk of celery and you can slice it or cut it in half, and you'll be able to see the vascular bundles. You can see them out here. And the lighter portion is the xylem, and the darker portion, or the little green portion there, that's the phloem. These are vascular bundles in celery. Pretty cool, eh? Okay, roots. Rhizoids of bryophytes don't do a very good job of absorbing water and nutrients or of anchoring the plant to the substrate, as you now know. Bryophytes, we're talking about mosses, but when we talk about terrestrial plants, we have to develop roots. Roots are vascular, as we've just seen, and they are able to penetrate soil and anchor land plants deep into the soil, so that when the first swift wind comes away, the plant doesn't blow over and like a tumbleweed, right? Roots allow plants to reach water sources that are unavailable to bryophytes, and roots also absorb nutrients from the soil. So this is probably typical of what you might imagine when someone says a root bundle. There you see the stem, the main root here, the accessory roots, and these little tiny roots are called adventitious roots. Here we see this little strawberry plant, just getting started. You can see the root bundle here under the ground. This is a row of corn. This is pretty cool. This is a row of corn, and you can see their roots, and corn stalks have very loose roots, and they don't go very deep. And you probably, hopefully, you've had the opportunity to go into a cornfield, or you've experienced this before. You can just grab a stalk of corn and you can just rip it out of the ground with one hand, because those roots don't go very deep, and they're very loosely associated. Anyway, those are roots of corn stalks, and some roots are even edible. Carrots and turnips and radishes, those are the kinds of roots that people typically eat. All right, leaves, sporophylls, and strobili. Oh my. So another major evolutionary advancement came in the form of leaves. Leaves are, again, vascular, as we've seen. They're better at capturing sunlight than stems, for instance. They're more efficient at conducting photosynthesis, and therefore they're more efficient in making sugars. And they are full of phloem. Remember, sugars phloem downward from the leaves to all the other parts of the plant. All right, so we have two different types of leaves. Some are tiny and some are big, and we call them microphylls. Does that make sense? Micro, they're little, small. We see those. They usually have a central vein, and we see those in the form of mosses. And then we have megaphylls, which are big leaf. Now megaphyll, big leaf, does not necessarily mean it's a 6-foot tall leaf, but it's just bigger than a microleaf. And it's multi-veined. We'll look at ferns here in a minute. Ferns certainly have megaphylls, or big leaves. Most vascular plants, in fact, have megaleaves. Your rose bush in your backyard, those are megaphylls. Elephant ears in your mom's garden, those are megaphylls. These represent the sporophyte generation. Most of what you see in the plant world out there in the biosphere is sporophyll, by the way. A sporophyte, by the way, sporophyte. Now, sporophylls are modified leaves. Sporophylls are modified leaves of sporophytes. Sporophylls make sporangia. Right? These are the reproductive structures of plants. And sporophylls are modified leaves, and they include things like cones, like from pine trees, pine cones, and hemlocks and cedar trees all make little cones. Modified leaf sporophylls include flowers, they're just modified leaves, and fronds like palm fronds are sporophylls. Strobili, okay, so pine cones are sporophylls, modified leaves. But pine cones have their own special name called strobili, and I don't care that you remember this, but strobili are the cones of conifers. Conifers are plants that make cones, like pine trees and hemlocks and cedars. These are specialized leaves that contain sporangia. They just give them a different name called, we call it strobili, they call it strobili. I don't call it strobili, I just say pine cones. And I just know that they're sporophylls or modified leaves. Okay, so leaves come in a variety of shapes and sizes, and I'm just here to show you some of the different varieties and sizes, palm fronds, of leaves. There's pine needles, and yes, pine needles are leaves. They're the leaves of conifers or pine trees. Needles are the leaves of conifers, and there's that 6 foot or larger leaf. We don't know how big this guy is, but we can maybe presume he's an average size male. That's one huge leaf. All right, so Polypodiopsida. These are your true ferns. True ferns are the most recognizable seedless vascular plants. There's about 20,000 species. They're the most advanced seed-bearing plant, most advanced seedless plant, most similar to seed-bearing plants. They inhabit moist environments. Still, we have spores here, so we need to have some water in the area. So the sporophyte is a dominant generation, and when you see a fern out there, you recognize it as a fern because of its fronds, because of its big wide leaves. That's sporophyte, 2N generation. Fronds are large leaves. They conduct photosynthesis, and they play an important part in reproduction, as we'll see. Fiddleheads are the little curled up tips of developing fronds. I'll show you a picture in a minute. I'm sure you've seen this like on the Discovery Channel or somewhere, if not in real life. If you want to see them in real life, go out into the woods in April and you'll see fiddleheads. But on the bottom on the underside, I should say, of fern fronds are sori. Sori are groups of sporangia. Sporangia make spores. And sporangia, with regard to ferns, are homospores. So you have sporangia that make male spores and sporangia that make female spores, okay? Not like the mosses where we were heterospores. Okay, so spores are carried by the wind and they germinate into gametophytes. Remember, gametophytes are always haploid. Gametophytes produce both antheridia, that's the male, and antleridia, and archegonia, that's the female structure. Fertilization occurs as sperms swim to the ovum. Again, we still need moisture here with regard to ferns, and that's why you see ferns in very moist environments. Lots of ferns in the tropics, right? They need water, still need water, even though they're vascular now. And fertilization occurs when sperm meets egg and a zygote is formed. A zygote, as you know, is 2N. And now we're back to the sporophyte generation. Alternation of generation. Sporophyte to gametophyte, back to sporophyte, or 2N to 1N, back to 2N, or if you want to go diploid to haploid, back to diploid. Alternation of generations. So ferns, fronds, and fiddleheads. These are just some images showing you some fern, fronds. You've seen these before. You're familiar with these. You readily recognize this as a fern. On the underside of ferns, now if you have ferns at home, or your mom has ferns in the house, you can turn the leaves over, and you will see these are sori, these little brown obelisk structures are sori, and those sori are full of sporangia, which make spores. Okay. And these are fiddleheads. I showed you this image at the very beginning of this lesson. These are fiddleheads, just immature or maturing fern fronds. They are edible, by the way. Not the ferns, but the fiddleheads. And here's the life cycle of the fern. Again, alternation of generations, 2N to 1N, sporophyte to gametophyte, diploid to haploid, however you want to look at it. Here's the sporophyte, which is a dominant generation for ferns. Those are the leaves that you see out there. You turn them over and you see the sori. They form, these are bunches of sporangia. Sporangia make spores. Spores are 1N. Spores germinate to make gametophytes. Gametophytes are 1N. They make gametes. So gametophytes are always 1N. So from the antheridia, you get, that's the male structure, you get sperm, 1N. From the archegonia, that's the female structure, you get eggs, that's 1N. Sperm meets egg, that process is fertilization, 1N plus 1N equals a 2N zygote. Now we're back to the diploid generation. Zygote grows into a new sporophyte. Now we're back to sporophyte generation. Okay, alternation of generations. One final video is from Kalana Harith about ferns. It's really pretty good. Not very long. Check it out. And one final quick check. Which defining statements about the fern life cycle is false? Sporangia produce haploid spores. Sporophyte grows from the gametophyte. Sporophyte is diploid while the gametophyte is haploid. Sori form on the underside of the gametophyte. Okay, so sporangia produce spores. That's always true, right? Sporophyte grows from the gametophyte, right? That's alternation of generations. Gametophyte to sporophyte, back to gametophyte. So that's true. Sporophyte is diploid, that's true, while gametophyte is always haploid, that's true. And sori form on the underside of not the gametophyte, but the sporophyte. So D is the false statement. That's the end. Any questions, let me know. And that's the end of chapter 25. See you in 26.